What's up, guys? Okay, so today we are going to be going over some drug action slides for medicinal chemistry. So this is an introduction to medicinal chemistry, or the chemistry of drugs. Okay. So the functional groups determine the way the molecule works, both chemically and biologically. So this is the center carbon, and these is an example. See, these are some of the functional groups on it. So some of the important things to note are some important terms such as pharmacokinetics, ADME, which stands for absorption, distribution, metabolism, and excretion. Pharmacodynamics, so is it on target, off target, anti-target, toxicity and side effects that can be caused by certain functional groups, and a bit about the patient. The patient is compliant, the patient's drug history, drug response, previous medical history, and their current medical history. So for medicinal chemistry, or medchem, for people who are studying pharmacy, these are some of the important objectives to note. Discuss the process and technologies involved in drug discovery, so those technologies including combinatorial chemistry, so this type of chemistry is also important, high throughput screening, fragment-based drug design, and database mining, etc. Stating the physiochemical properties related to biological action of drugs, so the pharmacophoric elements. Saying the concept of drug likeness, drugability, and Lipinski's rule of 5. Saying the concept of classical and non-classical bioisosterism. State the basics of structure-activity relationships. Discuss the basics of computer-aided drug design. Receptor-based and ligand-based approaches. Identify pharmaceutical salts and state the influence on important properties of drugs. Identify prodrugs and state the concept of prodrugs. State the categories of drug metabolism, so functionalization and conjugation reactions. Discuss the role of, for example, cysteine heme enzymes, the basics in drug metabolism. Discuss the concept of structurally based therapeutic evaluation. So these are some of the main points. So introduction to medicinal chemistry. What is medicinal chemistry? How does the drug approval process work? And what are some important medchem activities? So what is medicinal chemistry? The American Chemical Society division, according to the American Chemical Society, it's a division of medicinal chemistry. Medicinal chemistry is the application of chemical research techniques to the synthesis of pharmaceuticals. During the early stages of medicinal chemistry development, scientists were primarily concerned with the isolation of medicinal agents found in plants. Today, scientists in this field are equally concerned with the creation of new synthetic drug compounds. Possibly based on newly discovered mechanisms. The focus on development of new synthetic drug compounds has resulted in the incorporation of many other disciplines, such as biochemistry and molecular biology. Into medicinal chemistry. Medicinal chemistry is almost always geared toward drug discovery and development. So what is medicinal chemistry? Medicinal chemists prepare and or select appropriate compounds for biological evaluation that, if found to be active, could serve as lead compounds. They then evaluate the structure-activity relationships, SARS, of analogous compounds with regards to their in vitro and in vivo efficacy and safety. Today, medicinal chemists who are engaged in drug discovery are part of interdisciplinary teams and must therefore understand not only the field of organic chemistry, but also a range of other disciplines to anticipate problems and interpret developments to help move the project forward. So a lead compound is a chemical structure that shows activity and selectivity in a pharmacological or biochemically relevant screen. SARS, structure-activity relationship, is the correlation of structural features with the activity of compounds in a given assay. Apologize for the background noise. Those are people in the room who are speaking Filipino. Okay, so structure-activity relationship is the correlation of structural features with the activity of compounds in a given assay. MedChem occupies a strategic position at the interface of chemistry and biology. So what is medicinal chemistry? So these are some educational activities of medicinal chemists. So medicinal chemists can be involved in industrial internship, which some of the disciplines in that involve pharmacology, medchem knowledge, AdMet, clinical studies. So what some of you may not know what AdMet is, so I'm just going to define it for you here. So AdMet is an abbreviation in pharmacokinetics pharmacology for absorption, distribution, excretion, and absorption, distribution, metabolism, excretion, and toxicities. So I'm just going to put that here for you guys. So absorption, distribution, metabolism, metabolism, excretion, and toxicity. Okay, so that is AdMet right there. So, you know what, I'm just going to as well put like an arrow thing right here that it looks good. Okay, so they also work a little bit with clinical studies, and so yeah, all of these are part of knowledge that medicinal chemists should know. So part of their training for medicinal chemists includes computational chemistry, structural biology, synthetic chemistry, and working with, of course, natural products. Okay. So here's the drug approval process in the United States. So we start with, I'll start with this more generalized version before we reach the graph. So it starts, the drug approval process first starts with a preclinical stage. So a new compound is tested in vitro and also in vivo. So let's find out what that is for you guys. So in vitro is performed or taking place in a test tube, culture dish, or elsewhere outside a living organism. And in vivo is performed or taking place in a living organism. Okay, so let's just highlight that. living organism. No, wait. Outside a living organism. Just put it right here. In vitro. Should italicize that. Italicize. In vitro. And then there's in vivo. So in vivo takes place inside a living organism. All right. Just gonna highlight this real quick. It's very helpful. There we go. So preclinical stage is a new compound tested in vitro and also in vivo in laboratory animals for the evaluation of toxic and pharmacological effects / the investigation of genotoxicity. You start by filing an investigational new drug application, or an IND application with the FDA. And then you get a pharmacological profile and short-term toxicity testing in at least 2 animal species. And then you begin with phase 1, the clinical trials in humans after 30 days. So you start with a small number of healthy volunteers. You test for dosing range and toxicity. Next, you reach phase 2. For phase 2, you work with larger samples of volunteers or patients. And then for phase 3, you work with a larger sample of subjects, patients, with different dosing quantities or schedules to test for efficacy and any adverse events. And then you finish with the submission of a new drug application or an NDA. You submit your NDA to the FDA. Okay, now we're going to analyze this graph right here. So preclinical testing is right here. Starting with research and development takes about 1 to 3 years. Average is about 18 months. Initial synthesis, so there's some short-term animal testing. You hold for 30-day safety review. And then comes the clinical research and development stages. So this can take about 2 to 10 years, an average of 5 years. And during this process, you start working with your patients, or of human subjects. Phase 1, phase 2, and phase 3. This one, originally it was short-term animal testing, and this one is long-term animal testing because it's 2 to 10 years. And then you get your NDA review, your new drug application review. Once your NDA is submitted, it's reported to the advisory committee, and then the NDA, if you're lucky, is approved. Now the NDA review can take about 2 months to 7 years. It's a very long time. Average is 2 years. And then after that, your product is released, and then there's some post-market surveillance that goes on. So all of these are color coded. So the orange ones involve industry time, and the blue ones is for the FDA, Food and Drug Administration's own time. Okay. Now I'm just going to try and see I'll just leave it like that. Okay. So here's some important medchem activities. So medicinal chemists work for, or a lot of their main joy comes from this discovery program, and not the TV show, I mean through lead identification. From lead optimization to potential drug substance candidates and chemical pre-formulation. So all of this is all a part of architecture. Lead optimization, finding a new drug, to potential drug substance candidates. Important medicinal chemistry activities include, so defining that discovery program and lead identification process. First we have natural products or pharmacognosy. This can involve phytochemistry, and these are some other important approaches. Actually, I'm kind of curious what pharmacognosy is. Let's find that out. So, pharmacognosy is the branch of knowledge concerned with medicinal drugs obtained from plants or other animal sources. Okay, let's just put that in. So, I ate corned beef.
I know, how you didn't eat the egg?
So pharmacognosy is concerned with...
So can I eat it? Can you eat it?
Whatever.
You huh?
Whatever.
Are you gonna eat now?
In a bit.
Okay, I'm gonna eat it.
Pharmacognosy is the branch of knowledge concerned with medicinal drugs obtained from plants or other natural sources. Phytochemistry. So here are some other important approaches. So there is combinatorial chemistry, which was an important topic we discussed earlier. So, or it can be called solid phase reagents and scavengers. Another important thing, HTS, high throughput screening. SOSA, selective optimization of side activities. Structure-based drug design, including in silico screening. Fragment-based drug design, cheminformatics and database mining, using SciFinder, Scholar, NCL database, PubMed database, etc. Continuing with architecture. The architecture includes lead optimization, potential drug substance candidates, and chemical preformulation. So some of the topics for architecture include chemical space, drug likeness, druggability, structure, activity, relationship, SAR, and QSAR, drug target interactions, functional groups, pharmacophore, physiochemical properties, functional groups, and Lipinski's rule. Compound quality, drug quality, optical isomerism, bioisosterism, privileged scaffolds, hybrid concept, template concept, multi-target drugs, toxophoric groups, carcinogenic groups, prodrug concept, and bioprecursor, the blood-brain barrier. Chemical preformulation includes oral bioavailability, pharmaceutical salts, water-soluble compounds, and any formulation problems. Into the architecture, there is more on the discovery program and lead identification.
Hey, I'm gonna eat your food or what?
Yeah, go ahead. Which is mainly based on extracting compounds from natural sources ethnopharmacological sources. So what is an ethnopharmacological source? Okay. Doesn't really tell us. Okay, that's fine. Okay. Synthesis. So there's a synthesis example would be combinatorial chemistry. Compound collections, libraries, and databases also from libraries. So these would probably be ethnopharmacological sources. And the next part of the process would be the selection of suitable bioassays. Discovery program / lead identification listing without ranking. We have systematic screening, rational design, listing, okay, high throughput screening, HTS. So what is HTS exactly? High throughput screening. Okay. Doesn't look like this. High throughput screening is a drug discovery process widely used in the pharmaceutical industry. Leverages automation to quickly assay the biological or biochemical activities of a large number of drug-like compounds. Okay, let's use that. Okay, where is it? That is a lot of words. Able to shorten this. I'll be there. Okay. We have analog design right here. HTS, so against specific biological targets. We have, I'm just going to highlight this. Oh no, it's not possible to, never mind. Against specific biological targets, we have bioassays using compound collection, in silico screening, using databases, and HIT. So analog design takes part in modification of an existing active molecule for improved medicine, new intellectual property. So a bit of about rational design includes knowledge of the molecular mechanism and the disease related. Next would be selective optimization of site activities, or the SOSA approach. Which is used for testing known drugs on new pharmacological targets. Testing known drugs on new pharmacological targets. Next one is serendipitous observations, so unexpected clinical or pharmacological activities. What does serendipitous mean actually? Occurring or discovered by chance in a happy or beneficial way, or accidental. That's it for this video, guys. Thanks for watching.